This is Dr. Thomas Dayspring, Chief Academic Officer at True Health Diagnostics, with a tutorial on oxidized low-density lipoprotein. Here we see an overview of atherogenesis. What happens is ApoB containing lipoproteins, pretty much dependent on particle number, pass through the endothelial barrier and bind to arterial wall proteoglycans. While there, the surface phospholipids and free cholesterol are subject to reactive oxygen species. Once an ApoB-containing particle becomes oxidized, arterial wall macrophages, which originate from invading monocytes, develop scavenger receptors which internalize the oxidized lipoproteins. After they accumulate enough of them and digest them, they become what are called foam cells, which are basically sterol-laden macrophages. These macrophages secrete a whole variety of chemokines, cytokines, and inflammatory proteins, which further contribute to the ongoing chronic inflammation, creating further endothelial dysfunction. But, and the key point for this lecture is that those monocytes turned macrophages do not ingest ApoB particles, the vast majority of which are LDLs, unless they are oxidized. Let's take a close-up view of the anatomy of a low-density lipoprotein. Like all such particles, it has a surface layer and it has a core of other lipid molecules. On the surface, first we depict free cholesterol. You see its structure to the left, and in the cartoon it's depicted as a yellowish circle. Notice the cholesterol molecule has a hydroxy group on the first ring, and it has an aliphatic chain extending from its last ring. Phospholipids are the major moiety on the surface of all lipoproteins, and phospholipids contain two fatty acids, most often long-chain fatty acids, but they could be shorter chain in some circumstances. And of course, phytosterols, if present, also exist on the surface of lipoproteins. And take a close look at the structure of the phytosterol. This one would be cytosterol, and compare it to free cholesterol. They look identical. If you look at that aliphatic side chain, there's an extra ethyl group on cytosterol, and that's the only difference. The key characteristic of all of these lipid molecules that form the surface of lipoproteins is that they are what we call amphipathic. One side is water-soluble and the other side is not. And the water-soluble side can interface with plasma, which for the most part is an aqueous solution. Now, what's in the core of lipoproteins? Variable amounts of esterified cholesterol called cholesterol ester. And if you look at the structure, you can see there's a fatty acid attached there. The R would vary depending how many carbons are in that fatty acid. And of course, also present are triglycerides. Now, cholesterol ester and triglycerides are incredibly hydrophobic molecules, and that's why they are in the core of the particle and not on the surface. Now, this lecture is about oxidized LDL particles. All LDL particles, VLDLs and IDLs also, are held together by a large peptide, of which there is a single one per particle, called apolipoprotein B. And you're going to see which of these lipids and what happens to ApoB when oxidative stress is present. Which of the components of the LDL are subject to oxidative forces? Well, it's pretty much the molecules on the surface that have exposed oxygen moieties, or fatty acids that have double bonds in them, which would be the phospholipids. Those are the molecules easily affected by oxidation. Now, ApoB is a peptide, and oxidative forces definitely affect it and actually are going to change its shape, and we'll get into that later. we have to talk a little bit about atomic structure. And you see an atom staring you in the face. And of course, atoms are the smallest units of matter, and they define all of the elements in the periodic chart. And the vast majority of their mass is in the nucleus. It's the protons. Atoms also contain neutrons in its nucleus, but around the periphery are variable numbers of electrons. The simplest atom we have, its atomic number is number 1, is hydrogen, which consists of 1 proton and 1 orbiting electron. Let's take a look at carbon, crucial to human biology and all organic substances. And you can see it's a little bit different than the hydrogen atom. It has 6 protons and 6 electrons. And the electrons exist in these orbits. And you can see in the inner orbit there are 2 electrons, and in the outer orbit there are 4. That outer shell is called the valence shell because those electrons that are in the outer orbits can interact with other atoms. And of course, the electrons have a negative charge. Here you see a hydrogen atom and a carbon atom. The blue dots are called valence electrons in the carbon atom, and the red dot is called the valence electron in the hydrogen atom. That means those electrons are capable to binding to other valence electrons in other atoms. Carbon has 4 electrons in its outer shell or its valence shell. Hydrogen has 1. The presence of valence electrons obviously are going to determine the element's chemical properties and how well it binds or does not bind to other elements. In a single covalent bond, 2 atoms share a valence electron in that outer orbit. They become a shared pair. So let's look at methane, which simply consists of 1 carbon atom and 4 hydrogen atoms. And you can see each hydrogen atom is sharing with 1 valence electron on the carbon molecule. And so these 2 elements stay covalently bound to one another. And that's what a covalent bond is. You're sharing electrons that helps you stick together. Next, let's talk about oxidation and free radicals. And in the center of the graphic, you see the oxygen molecule. And notice there are 8 electrons, 6 of which are in the outer valence ring and 2 in the inner ring. And it's the electrons in the outer ring that are available to interact with other atoms. Oxidation in a chemical simply refers to the loss of electrons, usually in a reaction involving oxygen, but other molecules can do this. And it results in unpaired electrons in the valence or outer ring. Reactive oxygen species, which you often see written as ROS, are highly chemically reactive molecules that contain oxygen, and these have important roles in cell signaling and cell defense, but also in cellular toxicity. Examples include peroxides, of which there are numerous types, superoxide, and something called the hydroxy radical. You see in the diagram below is the oxygen molecule we breathe, dioxygen, and that is 2 oxygen atoms that are sharing electrons in that outer valence ring. They're covalently bound. And on the right, on the top, you see the hydroxy radical, oxygen and hydrogen, carrying an unpaired electron depicted in green, but it's also sharing an electron with hydrogen to which it's covalently bound. And on the bottom you see something called the superoxide. It's an oxygen-free radical or an oxygen-reactive oxygen species. And you see it's the dioxin, but the oxygen atom on the right is carrying an extra electron. These electrons depicted in green are available for binding to other molecules, and that's what the oxidative process is. A free radical is therefore an atom or a molecule or an ion that has unpaired valence electrons, meaning electrons in the outer shell, and that's called an open electron shell, and they can basically have what's called a dangling covalent bond. They're available to bond to another atom that has space for it. This makes them extremely reactive towards many other substances. And the half-life of these free radicals are a second. Boom, they interact virtually the instant they are formed. Here are depicted oxygen and some free radicals. Top left is the dioxygen molecule, where two oxygen atoms are sharing electrons in the valence ring. Bottom left, you see hydrogen peroxide, which is two oxygens that are covalently bound to hydrogens. And you see each hydrogen and each of the oxygen molecules are sharing one electron. Hydrogen peroxide, its symbol is H2O2. Now the others, take a close look. In the top middle is the superoxide anion. And you see it's two oxygen molecules, but they have an unpaired electron depicted in green. In the bottom center, you have what's called the hydroxyl radical. And that is an OH group, oxygen and hydrogen, but the oxygen is carrying an unpaired electron. So both the superoxide anion and the hydroxyl radical are imminently available to attack molecules that are subject to oxidation. Now on the right, on the top, you have peroxide, and you can see it's two oxygen molecules that have unpaired electrons available to interact. What's the difference between peroxide and hydrogen peroxide, bottom left? Well, in the bottom left, hydrogen peroxide, the two oxygen molecules are covalently bound to the hydrogen. But if those hydrogens pull off, boom, it becomes a peroxide, which is then able to bind to other molecules. And the bottom right is the hydroxyl ion, and it's pretty much the hydroxyl radical, except there is no unpaired electron. But it only takes inflammatory conditions, oxidative conditions, for one of those electrons to become unpaired. So the hydroxyl ion can become a hydroxyl radical very quickly. And notice what makes the radical. There's an unpaired electron in its outer shell. Whereas the hydroxyl ion on the right, all the electrons, at least temporarily, are paired. Let's take a close-up look at the oxidative processes might occur to one of the fatty acids that's part of a phospholipid on a cell membrane. So lipid peroxidation, which is a term that simply means oxidative degradation of lipids, and it's one of the many indicators that oxidative stress is going on in an afflicted human. Unsaturated fatty acids, typical of those in-cell membranes in the phospholipids, are one of the more common targets for free radicals. You see the chemical structure of such a unsaturated fatty acid. R1 would be extra carbon hydrogen atoms depending on the length of that fatty acid, and R2 on the right is typically what is called a carbonyl group. It's carbon bound via double bond to oxygen and another hydrogen. You see its molecular structure depicted both in graphic form and in nomenclature form. Now, the hydroxy radical, once it's formed, is looking to attach to another atom that has space for it. Remember, it has an unpaired electron, and this occurs extremely quickly after it's formed, less than a second. So, look at that fatty acid. You see the hydrogen depicted in blue there. Here comes the hydroxy radical with a free electron, which is colored as a red dot in this diagram, and it's going to interact, it's going to steal that hydrogen, and then you create water. But what happens to the fatty acid? You now have what's called a carbon-centered radical. The carbon atom now has an unpaired electron. The hydrogen atom has left and become part of a water molecule, which the body can easily get rid of. So the free radical captured the hydrogen moiety that was in that unsaturated fatty acid carbon to form water, leaving an unpaired electron on the fatty acid. And guess what? That's going to be readily available to bind to oxygen in the oxidation process. And what's going to be formed is something called a peroxyl radical. So oxygen sees that unpaired electron, rapidly binds to it, and creating a peroxide radical. And you see the chemical structure written on the bottom. Remember the structure that was on top? You can see all of a sudden there's now an oxidized molecule in the middle of that fatty acid. And that's going to subject it to virtually immediate degradation. It's not going to function structurally in the cell membrane as would the normal fatty acid, and that's going to change cell signaling and the entire integrity of the cell. And if this happens to too many fatty acids and too many phospholipids, that cell is probably going to die. Now, these lipid peroxides are rather unstable molecules, and they rapidly denigrate into other molecules, one of which is called F2 isoprostanes. Well, F2 isoprostanes are readily assayable in urinary specimens, and they are basically prostaglandin isomers that are formed in vivo from the degradation of oxidized fatty acids, especially arachidonic acid. Isoprostanes are pretty much formed by free radical catalyzed mechanisms. They are stable molecules. They are not produced by any enzymatic changes of fatty acid. It's oxidation that creates them. So F2 isoprostanes are specific biomarkers of lipid peroxidation. There are many classes, and it's the F2 class that can be easily assayed in the urine. And they have, in studies, been correlated very well as being present in the early stages of plaque formation. Because remember back to slide 1, atherogenesis is it results from oxidation of ApoB containing lipoproteins. Now, laboratories now can measure oxidized low-density lipoprotein concentrations. And in this lab report, you see the F2 isoprostane concentration, but LDL oxidative measurement is also abnormal. So what is the difference? Well, F2 isoprostanes is just a marker that oxidation is going on. But if the oxidized LDL biomarker is positive, you know some of those oxidative forces are attacking LDLs, which of course is very early in the atherogenic process. So now I want to better relate the oxidative forces into what they do to ApoB containing lipoproteins. And an imbalance towards the pro-oxidative state, which we have defined as the presence of free radicals, is also called oxidative stress. You see an LDL particle with a single molecule of ApoB has entered the picture. Interaction of oxidatively produced reactive aldehydes with the lysine residues. Lysine is simply an essential amino acid that is part of the ApoB peptide. In the ApoB molecule is going to change the qualities of this LDL. It's going to make it more negatively charged. Aldehydes are going to replace the lysine amino acids that are in the ApoB, and that's what causes changes in conformation. When you start changing conformation of ApoB, you will decrease its affinity to bind to the part of the LDL receptor that is looking to bind to apolipoprotein B. You will also increase the affinity of ApoB for scavenger receptors that are certainly present in those macrophages in arterial walls, but in other immune cells throughout the body, in virtually every tissue in the reticuloendothelial cells. So let's take an omega-6 fatty acid, linoleic, and certainly the one that's often expressed in immune processes, arachidonic acid. In oxidative states, there is catabolism of arachidonic acid into, we've already mentioned them, isoprostanes, but also into lipid species that are called reactive aldehydes. And other molecules are also present or possible from oxidation of arachidonic acid. So, what do these reactive aldehydes do? They bind to or replace the lysine moieties that are on apolipoprotein B. And that's going to change the configuration of ApoB, which, as previously mentioned, is going to affect its ability to bind to various receptors. Scavenger receptors in inflammatory cells and the LDL receptors, especially in the liver, intestine, or other organs that might be looking to clear LDL particles. Here is depicted the report form from True Health Diagnostics on how a reportage of oxidized LDL appears. And you can see there are optimal ranges, intermediate risk ranges, and high risk ranges. And oxidized LDL is seen in any oxidative state, and some of the more common ones are polycystic ovarian syndrome, other insulin-resistant syndromes such as diabetes and the metabolic syndrome, but other inflammatory diseases such as Alzheimer's disease, rheumatoid arthritis, or other autoimmune diseases, and of course, atherogenesis. Now, if you look at the bar graph on the right, where a study population where people are known to have variable degrees of atherosclerosis, and the Ox-LDL is divided into quintiles, there's a very steady increasing relationship. It looks to be directly related to the presence of coronary atherosclerosis. However, compare that to Ox-LDL versus LDL cholesterol, relating it to percentage of patients with coronary artery disease. The linkage, the association was much stronger with the oxidized LDL in a linear fashion, and it doesn't look like there's much relationship at all to LDL cholesterol. And we've known that from previous studies. That's why many of us use ApoB or LDL particle counts as a better diagnostic marker of atherogenesis than LDL cholesterol. Oxidized LDL is going to correlate with ApoB or LDL particle counts. How would it not? Because a certain percentage of the LDLs are likely to be oxidized, and the higher the LDL particle count, the more if there's an oxidative state present in that patient. Also understand that oxidized LDL concentrations are not particle numbers. They are simply referring to the presence or the absence of oxidized LDLs. So back to atherogenesis, and this is a similar graphic to the first slide of the lecture. And you see the ApoB circulating particles, you see monocytes in the plasma, and you see other factors that might affect LDL or ApoB particles, such as glycation or other fatty acids that exist in plasma as components of albumin. And of course, the LDL enters the arterial wall, it's subject to free radicals, it's converted to fully oxidized LDL, which is then internalized by the monocytes turned macrophages, creating the foam cells. And these foam cells secrete a lot of the chemokines and growth factors, etc. Now here's the key concept. The oxidative process where LDLs become fully oxidized occurs within the wall of the artery. It does not occur in plasma. So in essence, there are no fully oxidized LDL particles circulating in plasma. But yeah, we have an assay that measures oxidized LDL. But what the assay really measures is what is called minimally oxidized LDL. Any LDL that becomes more completely oxidized than in plasma is virtually instantly cleared by scavenger receptors in reticuloendothelial cells, and for the most part the oxidation does not occur in plasma because plasma is enriched with multiple antioxidants. Even many of the ApoB particles carry vitamin E. So the lab does not measure fully oxidized LDL particles. It measures circulating minimally oxidized LDLs. Now, as I've already showed you, such minimally oxidized LDLs, through a variety of processes, the ApoB changes conformation, which hinders the clearance. So LDL particles tend to accumulate. The oxidation allows these lysine molecules on ApoB to be replaced by aldehydes, which just is going to change not only the shape, but you're going to see when we talk about the assay, the laboratory uses monoclonal antibodies to bind to oxidized LDL particles or minimally oxidized LDLs, and those epitopes depend on the changing shape of ApoB. So let's talk about the assay, but a few reminders first. The oxidized LDL assays detect minimally oxidized LDL, which of course will have a correlation with more fully oxidized LDLs in the arterial wall, but you're not measuring fully oxidized LDLs because they really do not exist in plasma. If you look at oxidized LDL particles, minimally oxidized, and you compare them to all of the LDL particles that are circulating, they're a very minor fraction. In healthy persons, they pretty much don't exist. In people who are having an acute coronary syndrome, where there's many oxidative forces at play, maybe Ox-LDL totals up to 5% of total LDL particles. Small LDLs, which do contain lesser amounts of antioxidants, such as tocopherol, are more prone to reactive oxygen species than are the larger LDLs. Oxidized LDL concentration will always correlate a little bit with LDL cholesterol and certainly with LDL particle counts, but Ox-LDL is not a particle concentration and it's not a cholesterol concentration. We've already shown you oxidized LDL correlates well with the presence of atherosclerosis compared to LDL-C, and we've known for a decade that that is true with LDL-P versus LDL-C and plaque also. The oxidized LDL assay is an ELISA assay, and that stands for enzyme-linked immunosorbent assay, and it's what is called a two-site capture or a sandwich. You know, a sandwich has two slices of bread. One slice may be interacting with mustard and the other slice with, say, lettuce or what's on the other side of the sandwich. And that's exactly how this technique is going to work in the laboratory. So the first thing they do is they take a microtiter plate and they coat it with something called the capturing antibody. It's a 4E6 antibody, and it's a monoclonal antibody. And it's going to recognize the changed conformation of oxidized LDLs. And what's changed about it? The shape of the ApoB. And when you change shape of ApoB, the molecules to which an antibody binds, the epitopes, are different. So this capturing antibody will not bind to a non-oxidized LDL, but it will bind to the changed ApoB on oxidized LDLs. And here you see that binding occurring. The red and yellow lines extending from this LDL are meant to represent oxidized fatty acids. So you can see the monoclonal antibody is attaching to a certain segment of the ApoB, where those aldehydes have replaced the lysine, changing the conformation. Now, once this binding occurs, the lab does what is called washing. It washes away everything else that is not bound to this monoclonal antibody. What is the next phase of the process? Well, this is where the sandwich part of the assay comes in, because there's going to be a second antibody which is directed at other epitopes or amino acids on the ApoB, which have not been affected by the changed conformation of ApoB present in oxidized LDLs. And here comes that tagging antibody. And it's called a tagging antibody because it is bound to something else. It's conjugated with horseradish peroxidase. So this tagging antibody is carrying a peroxidase. And you can see the name of this tagging antibody. It's A82. HRP stands for horseradish peroxidase. And it is simply an anti-ApoB antibody, whereas the capturing antibody was specific only for the parts of ApoB that occur because the LDL is oxidized. This second antibody will attach to any LDL ApoB particle. But remember, we've already washed away the ApoBs that are not oxidized in the previous step. So now we have a captured oxidized LDL, which also is tagged with another antibody carrying a peroxidase. Now all the laboratory has to do is expose those bound LDLs with the peroxidase on them to a color-producing substrate called tetramethylbenzidine. And once the peroxidase reacts with this substrate, a molecule which creates colors and various degrees of color intensity is performed. And the more oxidized LDLs that are present, the more intense would be the colored reaction product. And that is easily measurable and then translated into concentrations which are reported to the clinicians. It's kind of a fascinating technique, and this is typical of what's called the sandwich ELISA. So now that you have oxidized LDL and you understand why you have them, you understand how they were measured, how can that be used in a clinical situation? Well, first of all, you might want to look for other inflammatory or at least markers of oxidative stress that might be present. And that would be, we've already mentioned, F2 isoprostanes. That requires a urine specimen. There are biomarkers that indicate endothelial dysfunction, symmetric and asymmetric dimethylarginine. Homocysteine is very much present in oxidative processes. The AspirinWorks test measures pathways of COX-1 and COX-2, urinary 11-dehydrothromboxane B2, and that is often associated with oxidative processes. And of course, having a deficiency of omega-3 molecules in the phospholipids of your cell membranes are associated with many ill health situations, including a propensity towards oxidation. How might one approach such a patient where Ox-LDL is abnormal? Well, like we do with everybody, you have to stop smoking. You have to put out the fire. The nutritionist would be key in such a patient to counsel them on choosing antioxidative rich whole foods, not supplements, which are not really proven to do very much, but the foods that supply a multitude of Mother Nature's antioxidants. Clearly, you'd want to control atherogenic lipoprotein particle numbers in these patients. Blood pressure control is essential to fight endothelial dysfunction and improve nitric oxide. Glycemic control in those where it's abnormal, or perhaps improving insulin sensitivity, should all be a way of fighting these processes. And of course, if there is a deficiency in omega-3s, supplementation or proper diets containing omega-3 acids, seem like a logical choice.